Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 36 of chapter 28, the applications of recombinant DNA technology. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. Application of the recombinant DNA technology can be used in all different aspects. So for example, in medicine, in agriculture and food production, in industrial uses, or in the environmental protection. A very well-known example of using the recombinant DNA technology in medicine is to produce human insulin. Human insulin can be produced using bacteria in large amount to control the blood glucose level in the diabetic patient, 糖尿病人. In the past, when we don't have enough human insulin supplies in the market, diabetic patients, they have to extract and get the insulins from the pancreas of cattle and sheep. So they are the mammals, so the insulin can be still usable but not very good in the quality because they does not belong to human insulin. The insulin from cattle and sheep, they might cause infection or very importantly, rejection in the human body. And this can be a very deadly problem. And nowadays, with the help of recombinant DNA technology, we can use this technology to produce insulin, the human insulin, using bacteria. And this is very important because we are producing human insulin. So this human insulin, as we know, it will not cause any infection or any rejection in the human body. And not only no rejection, it is also very fast in doing that, lower cost and large amount in mass production every time. So it has a lot of advantage in doing the recombinant DNA technology in insulin production. Let's take a look at the questions from your classwork 28.1. Question F, state two advantages of producing human insulin using recombinant technology over the traditional method of extracting insulin from the pancreas of cattle. So what are the advantages of doing the recombinant technology? That includes, for example, insulin produced by the bacteria does not cause rejection by the immune system, or a large amount of insulin can be produced in a shorter time than extracting from the cattle pancreas. Or the insulin is very pure and safer to use because the insulin extracted from the cattle, they might be contaminated with impurities. And last but not least, the extraction cost using bacteria is lower than extracting from the pancreas of cattle. So these are the advantages of doing the human insulin by the recombinant technology. In the medicine aspect, besides making insulin, gene therapy, 基因治療, is also a very well-known use of recombinant DNA technology. Gene therapy, it is a potential cure for disease caused by a defective gene, 有缺陷嘅基因. For patients who have genetic disease, they might have cells with some defective genes, such as defective genes to produce a malfunctioned protein. The functions of the protein is gone or the protein does not work as before. And now, with the help of gene therapy, we can use a vector 載體, such as a virus, to carry a normal gene, which will be able to produce a normal function protein. With the help of this normal gene, making a normal function protein, it helps to supplement the defective gene. Now, with a normal function protein, you can help the function and replace the function of the malfunction protein. While in the agricultural and food production aspect, the recombinant DNA technology helps to increase the productivity. You increase a gene which helps the GM, the salmon, to grow faster and grow bigger. Just like the case I have told you before about the 沙巴龍躉. A pest-resistant gene can be inserted into some GM cotton plant to increase the pest-resistant ability and also increase the productivity. So if there is a natural ordinary cotton plant on the right, it will be deteriorated and damaged very easily by the pest, 害蟲啦. But with the resistant gene, then the plant can grow very, very well. Insertion of a gene to increase nutritional value in some plant or crops is also possible. On the left, you see a picture of golden rice, and on the right, there are ordinary rice. What are their differences? Golden rice is actually a GM rice, and it is rich in beta-carotene, 胡蘿蔔素. This beta-carotene 胡蘿蔔素 can be converted to vitamin A inside our body. Since the rice is rich in the beta-carotene 胡蘿蔔素, it increases the ability to producing vitamin A in our body. And this is also increasing the nutritional value of the food produced. And vitamin A is very important for making visual purple in the rod cell, which helps to prevent night blindness, 夜盲症. DNA recombinant technology can be used in industrial aspect as well. For example, producing the stonewashed genes 洗水式嘅牛仔褲. For the details of how the stonewashed genes can be produced using the recombinant DNA technology, I recommend you to read it on your own. You can find the details in your textbook. Recombinant DNA technology can be used to help cleaning up the oil spills in the ocean. It helps for environmental protection as well. In the past, if there is oil spill on the surface of the ocean, one of the methods is to burn them by removing them using burning. However, this is not a perfect method because it leads to strong air pollution. With the help of recombinant DNA technology, bacteria with oil-eating ability can now be produced to help to clean up the oil spill on the ocean. Just for your information, nowadays, not only do we have the oil-eating bacteria for cleaning up the oil spills on the ocean surface, we actually have invented a type of sponge which helps to absorb oil on the water surface very quickly. So this is another method of helping environmental protection. So that's it for the lesson about the application of recombinant DNA technology. I will see you in the next lesson. Bye bye.